Hello students, once again welcome back to our biology class. Dr. Raghavendra here. Okay, in our earlier class we have discussed about the physiological importance of proteins, lipids, carbohydrates also. We studied even the proteins, how they are made up of, and their primary, secondary, tertiary and quaternary structures we studied, right? And the carbohydrates such as the polysaccharides, they can be the homopolymers as well as heteropolymers, how they are connected to one another, what is the position of glycosidic bond, yes, in the non-reducing as well as reducing chains of the polymers we have discussed. Now, in today's class, we will be studying about the physiological importance of nucleic acids, structural details of the nucleic acids we will discuss, as well as the dynamic state of metabolism as well as the dynamics of the metabolism and then living state we will be discussing. Fine. So here let us go ahead with mainly the physiological importance of nucleic acids, right? So nucleic acids as such, what they are? They are the most important chemical components that can be seen inside the nucleus as well as in the cytoplasm. So when you talk about, you might have been heard about a concept as such, central dogma of the cell. Central dogma of cell means whatever happens inside the cell depend on one important reaction, one important the path or the mechanism, what we call central dogma of the cell, what it is. So as such, in the cell we find the DNA, deoxyribonucleic acid. The DNA going to make its own copy multiple times. We call it as this mechanism is known as replication. So making its own replica or copies, we call the replication. DNA going to form the RNA. So ribonucleic acid. So this is called as transcription. Okay. RNA going to form the proteins, right? Proteins, what we call it as a translation, right? So the entire cell, yes, depend on these three important reactions: replication, transcription and translation. So any type of character has to be expressed in our body. Yes, it may be the height of the person, it may be the texture of the hairs, it may be the color of the person. All these are called as morphological appearance. It depends on the type of genes, the type of DNA we have, right? So when DNA is making its own type of DNA, this keep on happening inside the cell. So that mechanism is called as replication, and this further the DNA going to form the RNA. DNA basically it is a double-stranded structure. DNA double-stranded structure. RNA is a single-stranded structure, and protein is what we can say. Yes, it is the long chain of these are the amino acids. Okay, long chain of amino acids. So anything that can happen in the body means increase in the height or growth or formation of particular tissues that require the protein. Yes, for the growth protein is required, for metabolism enzymes are required, for the immunity or defense mainly the antibodies are required. For all this, instruction coming from where? Instruction or the command is coming from the DNA. So DNA is making its own copies, replication we say, DNA making RNA, it's called as a transcription. So RNA making the translation, the protein formation means any type of protein has to form, tissue has to be grown up, or a character has to be expressed, all depend on the DNA. We call it the central dogma of the cell. So this is the physiological importance of what we can say the nucleic acids, what we say the DNA and RNA. Yes. Who first time reported yes there is the existence of the DNA? Let me tell you, mainly somewhere in 1954, yes, it is the Crick and Watson, yes, developed they developed DNA double helix model. DNA double helix model based on reports, okay, of reports provided by Wilkins. Okay, so what is these reports? So actually it is X-ray crystallography X-ray crystallography developed by Rosalind Franklin. Yes. So we people know always the Crick and Watson, but we don't know who are the actual worker behind the development of the DNA double helix model. It is mainly the Rosalind Franklin. So somewhere in 1962, yes, for the formation for the giving this DNA double helix model, they are rewarded with the Nobel Prize. So 1962, yes, the Nobel Prize shared by Crick, Watson and mainly the Wilkins for developing DNA double helix model. DNA double helix model. So why not Rosalind Franklin? Yes, rewarded, because till that time, yes, only Wilkins, the guide who guided the Rosalind Franklin to do the X-ray crystallography. So only the Wilkins was alive. Okay, so Rosalind was exposed herself to many more times to the X-rays and she died with that. So the misery behind the discovery of the double helix model, yes, it is very, very tragic. We can say that actual discovery is mainly the Rosalind Franklin. Well, so let us go ahead. What actually the DNA has? So we need to understand all these things basically. Yes, the central dogma of the cell means it is mainly the DNA that is going to form the RNA, and then it is what we can say that proteins. So the 1954, yes, the development of the double helix model. 1962, reward for that double helix model of DNA. Fine. There is a comparison. Yes, you need to understand, you need to know what is that comparison. Yes, the comparison between DNA and RNA is very, very essential and important. So when we talk about yes, the DNA as well as RNA, before we go into the details of the DNA structure as well as RNA structure or the types, yes, let us understand what are the basic things. So DNA, it is a double helix. RNA is a single strand structure, synthesized in nucleus and remains in nucleus. Yes, produced in nucleus, but okay, yes, perform the functions, but become active in the cytoplasm, but become active in cytoplasm. Then the first difference, second difference. Third, nitrogen bases. Nitrogen bases are adenine, thymine, guanine, cytosine. Nitrogen bases. Nitrogen bases are adenine, uracil, guanine and cytosine. No thymine. Thymine is replaced by the uracil. Then number 4, highly stable to temperature and pH, as well as the pH fluctuations it can tolerate, whereas RNA is sensitive, can't resist the fluctuations of the temperature and pH. Number 5, has self-repair system. So, no self-repair mechanism. Independent for its own synthesis. It is dependent on the DNA. Sixth difference. Independent synthesis. Okay, dependent on the DNA. So synthesis. Okay, so these are a few basic differences. It is double helix, it is single strand. It is having mainly the synthesized in the nucleus. That is also nucleus, but remain active in the nucleus, but it is remain active in the, what we can say, cytoplasm. Nitrogen bases ATGC, nitrogen bases are AUGC. Highly stable, self-repair mechanism, then tolerates the temperature and pH fluctuation, but it can't tolerate. So, self-repair mechanism, no self-repair mechanism, independently synthesized, whereas that is dependent on the DNA only for synthesis. Now, one more important difference. The sugar is deoxyribose sugar. So here the sugar is ribose sugar. It has a ribose sugar. So these are few important and basic differences what we can find in the DNA as well as RNA. Fine. So that is about the differences what we have studied. Yes, I hope you got a clarity about this. Mainly when you talk about the central dogma of the cell as well as DNA and RNA differences. Well, one more important thing. Yes, mainly being a single stranded, it is a double stranded, but the, what you can say, polymorphism can be seen in the DNA, RNA. What is that? So yes, it has three forms. What are they? They are mainly what we call as the mRNA, yes, tRNA, rRNA. mRNA is very less, least percentage, 3 to 5% only among the total. tRNA 10 to 15%, or sorry, 10 to 20%. Whereas ribosomal RNA is yes, dominant in terms of what we can say percentage wise, it is 80%. So we can say that there are the three forms of RNA, whereas the DNA is a single type, but it can be, we can say that zDNA as well as bDNA. Yes, well known types is zDNA and bDNA. bDNA is right-handed, so zDNA is left-handed. So we can say that bDNA means yes, it is right side winding, right-handed, and zDNA is left-handed. Means when we are talking about, yes, structural details of the DNA, you will find spiraling. So it is mainly spiraling to the right side. That is called the BDNA. Spiraling reverse, then it is the left-handed ZDNA. So these are the few basic differences. Yes, we need to know. Yes, what are the basic differences between the ZDNA as well as yes, the BDNA or the RNA forms. These are all the three types of RNA. Well, so let us go ahead. Yes, mainly regarding the structural details of the DNA also. As we know that DNA, yes, what actually it has? DNA has base pairs, we call them. So DNA is a polymer of nucleotides. So DNA being a polymer of nucleotide, let us understand how actually the nucleotides are arranged. Yes, we have seen, and we have to understand mainly phosphodiester bond, what actually and how it is mainly arranged or found. So as such, DNA, we say that yes, the polymer of nucleotides. Nucleotides, we know the nucleotides are adenine, guanine. Yes, we say that adenine, guanine is what? These are the purines. Then thymine, cytosine is mainly pyrimidines. Now, when we talk about yes, mainly in the DNA, yes, DNA is a double helix like this, and a small part, yes, we are going to take it, and we are going to study that. Yes, so here adenine combined with the thymine, and there we find mainly the sugar. Yes, here also we find the mainly the sugar. Then guanine, yes, with the 3 hydrogen. In the earlier class we have studied about this. The sugar, what we find here is a deoxyribose sugar only. Yes, we see this, and everywhere the terminal oxygen, and then here it is the first, second, third, fourth and the fifth carbon. So first, second, third, fourth and fifth carbon. Here also the first, second, third, fourth and fifth carbon. The first, second, third, fourth and fifth carbon. So this fifth carbon and this third carbon connected by the phosphate group. Here also this third carbon and fifth carbon is the phosphate group. So we call it as the phosphodiester bond. Generally, what is this phosphodiester bond? So all you know that this is a glycosidic. These are the hydrogen, and here is the phosphodiester. So these are the different bonds that can be seen in the DNA. So mainly the phosphodiester bond, yes, it is a phosphate group with the OH here, double bond O here, one O is here, one O is here. Further it is connected with the next CH2 group of the next sugar. Okay, so yes, what we say that mainly it is, yes, the sugar where it is connected. Yes, here also it is mainly what we can say that this is mainly the fifth sugar, whereas here it is yes, sorry, here it should be mainly to the third sugar, and yes, here it should be with the fifth sugar. Okay, yes, so the fifth sugar, and here is the third sugar. So it is what we say that phosphodiester bond or orthophosphate group. Orthophosphate, and it's going to form the phosphodiester bond. Okay, going to form the phosphodiester bond. What you are observing. So thus always we can say that it is a 5' to 3'. So this you need to remember basic structure of a DNA and then how they are connected. Well, so some more details of the DNA let us understand. What are those further more details? Let me give you. Some more details of the DNA includes double helix structural details. What is that double helix structural details? Yes, we need to understand. A DNA definitely a double helix and the strands are anti-parallel to one another. Well, so when we talk about the DNA, double helix model of the DNA. So let us write the title double helix model of DNA. So when we talk about double helix model of a DNA, let me mention here 1, 2. Yes. So it is the 5' end and here it is the 3'. Here it is the 5' and here it is the 3' end. So the 1, 2, 3, 4 and 5. 1, 2, 3, 4 and 5. So this entire the loop, yes, it is called as one helix. So one helix is 34, what we can say that mainly it is 3.4 micrometers. Yes, 34 micrometers. Sorry, it is 34 angstroms. Yes, and then one helix has, we can say that 10 base pairs. BPs means base pairs. So now this entire length is 34 angstrom, but what is the, we can say this, it is a 3.4 angstroms. So the 10 base pairs, yes, they rotate 360-degree angle. What we are observing. One more important thing, mainly the diameter of the DNA. Yes, it is 2 nanometer or 20 angstroms. 20 angstroms. Yes, the diameter. And as such, always we can say that adenine pair with the thymine, guanine pair with the cytosine. Okay. So this is what we say about mainly the DNA is spirally, and then these strands are anti-parallel strands. Anti-parallel strands. Yes, one helix has 10 base pairs. Okay, we call it as a base pair. So base pair. So 10 base pairs. The distance between the base pair is 3.4 angstrom. The distance between base pairs. Okay, 34 angstrom is the length of one helix. The width of the DNA is 20 angstrom. Okay, adenine always, we can say that one more important thing, Chargaff's rule. Chargaff's rule. What it says? Sum of purine always equal to the sum of pyrimidines. Pyrimidines means A plus G always equal to P plus C. This you need to remember. Okay. This is the entire, the whole double helix model of the DNA. What you are observing here, it is mainly the double stranded DNA, and the double stranded DNA it has a backbone, what we can say, yes, sugar and phosphate groups are the backbone of the DNA. Sugar, what is that? Deoxyribose, oxyribose, and phosphate groups forms the backbone of DNA. Yes. And then one helix is mainly comprises of 10 base pairs and the length is 34 angstrom. The difference between one to the next base pair is 3.4 angstrom. Width of the DNA is 20 angstroms. Okay. So thus what we can say that both the strands run anti-parallel to one another. One strand go down, other strand come up anti-parallel to one another. And then always a purine pair with the pyrimidine only. Yes, we can say that always a purine pair with pyrimidine. So specific, you have to use the word specific purine pair with the pyrimidine, means adenine always pair with the thymine, guanine always pair with the cytosine. There is no adenine, cytosine or guanine, thymine will never happen. If it happens, it is called the point mutation. It is an abnormal status. That will never happen. So yes, we can say that DNA being a double helix, it has mainly the spiral, one complete turn, we call it the spiral, one helix. So one helix means 360-degree angle turn. So one helix, okay, one helix equal to 360-degree turn, 360-degree angle turn. So the angle difference between one base pair to next is 36-degree only. How? Because there are 10. So what happens? It is a spiral, right? It is a spiral, and on an angle you are observing, yes, like a staircase. Yes, it is mainly turning like this. So what happens? The 10 base pairs means 360-degree angle turn. One to next is only, it is 36-degree angle turn, or the difference is there. So these are all what I have mentioned here, most important and essential properties of the DNA you need to remember. So let me tell you once again, DNA is the most important nucleic acid that has the deoxyribose sugar, and DNA is a double-stranded structure and it has the ability to withstand against the fluctuation in the temperature as well as the pH, right? And then DNA has the ability to go fast and very fast recovery or self-repair, because one-sided any nucleotide are replaced or damaged, it has a what you can say template. It will guide for the arrangement of one more new thymine. So it will guide maybe to arrangement of the one more guanine. Like this, very fast the self-recovery will happen. So because of its double helix nature, we can say. And then, so arrangement always the specific purine pairs with the specific pyrimidine, adenine always thymine, guanine always with cytosine. Now, yes, we can say that one nitrogen base and one sugar is a nucleoside, and when it is a phosphate group, then it is a nucleotide. So here we can find mainly hydrogen bond between the nitrogen bases, glycosidic bond between the what we can say mainly the sugar and nitrogen base, and then phosphodiester bond between the sugar of adjacent or one after the next. So this phosphodiester has an orthophosphate, we can say that, and the fifth carbon, then phosphate group attaches with the third carbon of the next. So that's why it is a 5' to 3' always growing direction. So one helix means a 10 base pairs and its total turn is 360-degree angle. Difference between one pair to the next pair, angle difference I am telling, that is 36-degree only. So total length of one helix is what? Total length of one helix is basically it is 10 base pairs and then 34 angstrom. So distance between one to the next base pair is 3.4 angstrom. The diameter of the DNA is mainly 20 angstrom, we can say. And then these strands are anti-parallel to one another, and these base pairs are called complementary base pairs. Means a particular mainly the purine pair with a particular mainly pyrimidine. So adenine always pair with thymine, guanine always pair with cytosine. Adenine thymine has a 2 hydrogen bond between them. Guanine cytosine has a 3 hydrogen bond between them. According to Chargaff's rule, it says that sum of the purine always equal to sum of pyrimidine. Means adenine plus guanine is equal to thymine plus cytosine. According to this Chargaff's rule, you need to understand one more important thing. What is that? So always A plus G plus C plus what you can say T is equal to 100%. This you need to remember. So that's all about the structural details of the DNA also. In the next class we will discuss some more interesting concepts. Till then, take care and study well. Thank you very much.